Wunyangulum. Welcome everybody. In particular, I would like to welcome all of you who are HDR students who've chosen to come to be a part of this special event today. My name's Mark Orams and it's my privilege to serve as your Dean of Graduate Research at the University of the Sunshine Coast. An event like this is one which is always a joy to be a part of because what it does do is it provides us insights into some of the wonderful, diverse and important research that's going on across our university with our over 450 students who are doing higher degrees by research. In particular, I'd like to welcome our 8 finalists for the 3MT competition this morning. We are already very proud of you, as we are proud of all of our HDR students across USC. The challenge, as you will appreciate, of distilling what is often many, many years of work and something which is usually presented in a thesis which is somewhere between 50 to 100,000 words in a few short minutes is not an insignificant one. So we are all full of admiration for the challenge that you've taken on, and we look forward to learning about your research during your presentations this morning. I'd also like to acknowledge and welcome visitors to the campus, in particular our team from Unibank, who are our sponsors for this particular 3MT final, and I'll introduce you to Mike Lansing in a few minutes' time. As we get started, it's appropriate for us to acknowledge the Indigenous people of this place, the Kabi Kabi people. And so on behalf of all of us, I would like to acknowledge the elders from past, from now, and also elders who are yet to come in the future. We recognise that the space that we occupy has significance and importance for them, and that we acknowledge and respect that significance and importance. So I'd like to invite now Mike Lansing to come up and introduce himself and to let us know why this particular competition nationwide and indeed across the South Pacific is one which is so important to him and his team. Mike has come all the way from Sydney today to be with us, and he has members of his team who've travelled up from Brisbane to support this competition. So please join me in welcoming Mike Lansing.
Good morning, everybody, and thank you, Mark, for that lovely welcome. Look, Unibank is a mutual bank. It's owned by its shareholders. We're not paying dividends to shareholders. Our shareholders are our customers, and for one of the really key things about Unibank is its support for social responsibility. So things like the 3-minute thesis, which I saw for the first time at UQ a couple of years ago, was just brilliant. I mean, the enthusiasm, the passion that students have in relation to their thesis is just something. It was a highlight for me when I first joined Unibank. So it's a privilege to be here. It's great to be able to support a number of 3MT events across Australia. We do, I think, about a dozen. We're also the Asia Pacific sponsor for People's Choice. And research is absolutely paramount to Australian society. The consequences of what a lot of you guys do will benefit society as a whole. So I think for our social responsibility obligations, as we sit at Unibank, I think it really dovetails well with that. So, look, I don't want to say a lot more than that. It's great to be here, and I'm really looking forward to hearing the 8 contestants this morning. So good luck to every one of you, and have a great morning. Cheers.
Thank you very much, Mike, and thank you to you and your team for your support of the University of the Sunshine Coast today and through this 3MT competition. A little bit about the competition overview. Interestingly, some of you will have picked up that my accent is not from here. It's a Kiwi accent, and I first was exposed to the 3MT at a university in New Zealand, so it has quite significant reach. The competition has been underway for almost or just over 10 years now and developed locally here by the University of Queensland, but it has significant uptake across the university sector. So it's something that has been seen to be of value, both for the students involved in the challenges and opportunities it provides, but also I think for the wider university community and beyond, as people are able to learn about the exciting work that HDR students do across universities, not just in Australia, but across the South Pacific and internationally beyond there as well. So this competition is one which is judged, and it is judged on the basis of a series of heats that we have already held. And so today what we have is our 8 finalists who have been deemed to have performed extremely well, not just in terms of their presentation skills, but in terms of the substance and significance of the research that they're undertaking for their degrees. So you'll learn a little bit more about that during their presentations in a few minutes' time. So I'll introduce you to our judges for this morning. Mike, you've already met, and Mike has some experience now in judging at other universities as well as ours. And I'd also like to welcome Professor Roland DeMarco, our Deputy Vice-Chancellor of Research and Innovation, who's also serving as a judge this morning. Professor Marianne Wallace has not been able to join us, and so we will just have Mike and Roland serve as our judges through the competition. The rules the students who are presenting are quite familiar with, but they're up there just for you to be aware of. It is something where they must stop. You will not see it from where you're sitting out in the audience, but sitting in their eye line here down in the front row is a large iPad with a countdown timer on it. And so that countdown timer means that when they reach that 3-minute spot, they must stop. So they have practiced, I'm sure, and have worked through to ensure that their delivery meets that primary requirement. There are some important prizes, not the least of which is some financial incentive for the student who is both runner-up and also our winner. And those financial rewards are things that can be used to support their ongoing research with regard to their degrees they are pursuing. In addition, the winner is supported to attend and to participate and compete in the Asia Pacific final. That's going to be happening in October, and that's something that we hope that we know that our winner will represent us with a great deal of pride and do extremely well in that competition for us as a university community. The University of the Sunshine Coast is growing rapidly in the HDR space. We have grown somewhere close to 40% in our enrolments in masters and doctorates by research over the past 3 years. And that's a really exciting time to be a part of the university, and for those of you who are students in this area, to be a part of a growing institution. What you will find when you listen to these presentations is how remarkable the range and diversity of research is that's been undertaken. And this, of course, is only a small representative of the range that's going on across the university with those 450 plus students. So it's an exciting thing to be a part of. It's challenging, as it should be. It has significant obstacles to overcome, as those of you who are going through the HDR journey, and of course those academic staff who've been through it themselves will well recognise. But at the end, it's a very worthy application of your dedication, your intellect, your talent to achieve your higher degree by research. So it is indeed a privilege to be a part of it, and I would now ask you to just welcome our first presenter this morning. So Nicola Kent, thank you for being the first presenter this morning with regard to your presentation, which is entitled The Adaptive Diversification of the Eastern Water Dragon, and subtitled City Parks as Theatres for Rapid Evolution. Nicola is part of our Global Change Ecology Research Group, and she is based in the School of Science and Engineering here at the University of the Sunshine Coast. Nicola, we welcome you for your presentation. Best wishes.
Thank you. Yep, ready to go. Hi guys, so I'd just like to start by introducing you to the godfather of all things evolution, Charles Darwin. You probably know his work. So Darwin taught us many things, but mainly that species are not immutable and static forms. So his ventures on the Galapagos Archipelago in particular taught us that different habitats can drive divergence in species traits. So what we now know is that it's not the Galapagos in particular, but islands in general that create a pageant of natural experiments for the study of evolutionary processes. So if we skip forward 200 years-ish, what we're now seeing is that urbanisation is creating islands, contemporary islands, in the form of city parks and urban green space. So what we now know is that wildlife that inhabit these islands are experiencing changes to their phenotype. So for instance, changes in body size, body shape, stress physiology and behaviour. So what we are yet to understand is how urbanisation impacts the evolutionary processes linked to these changes. So my PhD is to better understand how natural selection can operate in an unnatural environment. So as Darwin said, it's not the strongest that survive nor the most intelligent, but it's the most adaptable to change. So for my work, I've chosen an iconic urban adapter, the eastern water dragon. These guys not only survive on these urban islands, but they thrive. So what this work has done thus far is to identify if geographically close, yet isolated populations of the eastern water dragon are experiencing changes to well-known evolutionary processes. So preliminary results are indicating that each individual urban island is experiencing selection on different morphological types. So what that means is on some urban islands, it's favourable to have longer legs, whilst others bigger jaws. So I'm now expanding on this research to look into the genome of the water dragon. So what that means is the genetic makeup. So here I'll be identifying markers that can be experiencing the same level of divergence on these urban islands. So these markers can be linked to functional genes that, as with longer limbs or bigger jaws for instance, may be favourable in certain habitats. So this work not only utilises these urban islands, comparing them to their native counterparts, but also looks at natural history specimens from museums to track this change through time. So all in all, this work will allow us to form an understanding of how urbanisation impacts evolutionary processes and can therefore be mitigated to create resilient and sustainable urban ecosystems. Thank you.
I'm always going to be wanting to hear more out of these presentations. I came out of that thinking, well, that's really interesting. Now, both personally from somebody who has relatively short legs and quite a big jaw myself, I kind of related to your presentation there, Nicola. I don't know if mine's an adaptive diversification, but anyway, sometimes we're just given the physiology we're given. Our next presenter is Chin Dam, and Chim is going to speak to us about Here Comes the Kingfish. And Chin is part of our Genecology Research Centre, also from the School of Science and Engineering. So please join me in welcoming Chin Dam.
I came to Australia to be a chef for Australian king. Some of you might ask, does Australia have a king? Well, I have the answer in the picture behind me. It is a kingfish, a rising star in aquaculture sector, mainly because it grows fast and is perfect for sushi and sashimi. In order to increase the production and profitability of kingfish industry, feed formulation is really the key because fish feed are expensive. Unfortunately, kingfish eat like a king. Their diet still rely on fish meal as main protein source. It makes this species more expensive to produce. This concern prompts a search for alternative plant protein source. However, the extensive use of plant is restricted because they contain anti-nutritional factor that may have a negative impact on nutrient digestibility and fish health. Or in other words, we don't expect our king to go vegan anytime soon. So my thesis is aiming to investigate the potential use of plant in future kingfish diet. Specifically, I'm focusing on how this alternative impact on nutrient digestibility, gut microbiota, and digestive enzyme expression. The way we do it was by feeding the fish with diet contain different plant protein source, and then we collect fish poo and gut tissue for nutritional and molecular analysis. So what we found is that our king can use faba bean meal, gluten canna meal, soybean meal as fish meal replacement without any negative impact. Surprisingly, our king need gluten-free diet because when they ate corn gluten meal, they were not able to digest it. And it's correlated with the reduction in the number of beneficial bacteria, and also it inhibits digestive enzyme gene expression in their gut. To summarize, my PhD so that we can use our understanding on the impact of alternative as a tool to formulate the cost-effective diet for kingfish and make this species to be a best sustainable Australia seafood in the global market. Long live the king. Thank you.
Thank you, Chin. I'm always full of admiration for anybody who can stand up in front of a group and speak in a language that's not their first language. So congratulations to all of you who take that on. It is something that is very difficult to do and something which I think all of us recognise as challenging and we're appreciative of. For our next presenter, I'm just going to do a quick mihi. It's appropriate because of the topic that she's introducing, for Hayley Whitfield, who is from our School of Education, and she's going to present on perspectives of life, learning and identity in a rural gang community. For those of you who don't know, a mihi is a greeting in Te Reo Māori, the Indigenous people of New Zealand, and this is appropriate because this is what her study focuses on. So Hayley, tēnā koutou katoa, ngā mihi nui o te mātaronga Māori o te Aotearoa. So welcome, haere mai, and we look forward to your presentation. Kia ora.
Thanks. Gangs. When I say that word, what are the first thoughts that pop into your mind? For most of us, our perspectives and views on gangs are formed from media, TV, music, probably not the most reliable of sources. And this is the guts of the research problem. We really know very little about New Zealand gangs, despite New Zealand having one of the highest gang member perspectives, gang member per capita populations in the world. We do know our gangs are different from other countries. Median age 40, predominantly Māori, stay in the gangs for decades and are dispersed across urban and rural locations. For example, Auckland, 1.6 million people, 19.5% of New Zealand's gang population. My research location is a small collection of towns and cities, 250,000 people, 18.5% of New Zealand's gang population. So there's been a call for more localised research, in particular things like the link between gang and whānau, or family. Why are gangs continuing to grow and flourish in New Zealand? Why are people staying in these gang subcultures and gang geographies for so long? And how does the intergenerational nature of this impact neighbourhoods? And why is there so little gang member perspective in the literature? Why do we know so little? Well, there's a lot of reasons, but one of the main ones is gangs are notoriously hard to research. They are secretive, they mistrust outsiders, they even have this saying, they say, ko te haina, ko te korero, don't sign, don't talk. Don't sign, don't talk, that's kind of a problem in the research world, right? So I find myself in this unique position where I'm allowed to research a gang community. My study then draws on subcultural theory, which is the study of non-normative social groups in society. And it's about trying to address this need for more localised gang research. It's also about giving gang members the opportunity to express their perspectives. And I'm using phenomenology, which is the study of lived experience. And the research question then is, what are Māori gang-connected adults' perspectives of life, learning and identity in a New Zealand rural gang community? And honestly, I've done about 30 hours of interview so far, and I'm really just in the data analysis stage, so my findings are very preliminary, but they are so interesting. Our gang members have talked about the gang, brotherhood, values, loyalty, honesty, trust, fatherhood, the need to be a good dad, a good husband, domestic violence, trying to overcome that challenge, their childhoods, poverty, rejection, learning, being illiterate, being alienated. They've talked about abuse. They've talked about abuse at home, abuse in the community, abuse from the government system. Who can you trust? You can't trust anyone but the gang. Mothers have talked about the need to raise the responsibility even, to raise their sons as good gang members. And recently a young gang member said this to me. He said, gangs, the world is so ignorant of us and we are so ignorant of them. Stop. Ask a question. You'd be surprised by the answer you get back. Thank you.
Kia ora, Hayley, ngā mihi nui. As a New Zealander who was born in a small town called Waitara in Taranaki, I can relate to a number of the things that you shared. So, as I mentioned at the beginning, what's fascinating about coming to an event like this is you learn the diverse range of really significant and exciting research that our HDR students are involved with, from creating more sustainable sources of fish for us to eat, through to examining the role of gangs in an Indigenous people in a neighbouring country, through to looking at neighbourhood parks and what happens with an important species and their adaptation to living in an increasingly urbanised world. So it is a fascinating space that we're all privileged to be involved with here at the university and the HDR community and the research that's undertaken. The next presentation is no exception. We changed gear again to look at something which is increasingly important for human experiences, particularly as we engage in our world more and more via social media. Angela Hintz is our next presenter. She comes from our School of Social Sciences, and her presentation is entitled From Social Media to the Yoga Mat. Please join me in welcoming Angela.
There's a meme going around at the moment, you might have seen it, and it says, how to get a yoga body. Step 1, have a body. Step 2, do yoga. That's it. And in the 12 years that I've been teaching yoga, I truly have seen it benefit women of all shapes and sizes, ages, and physical capabilities. However, let me introduce you to the yoga body as determined by print and digital media, where the diversity I've mentioned is not being reflected. The first stage of my research has involved me looking at how women's yoga is being represented on Instagram. I've coded close to 1,000 posts, and what I've found is that 80% of women in the posts were of either thin or athletic build, while only 19% of women displayed average levels of visible body fat. Only 1% of these women were overweight. 70% of the women were in their 20s, 75% were white, 100% were able-bodied. There are around 200 contemporary yoga poses to cater for all levels. However, in the posts I looked at, 50% of the poses were highly advanced. Many of them displayed unsafe alignment in the pursuit of aesthetic goals. So why does non-diverse representation matter? Well, we already know that exposure to idealised images of fashion and fitness models has a negative effect on women's body image. Not only that, when exposed to unattainable depictions of health, women tend to respond by either not engaging at all or engaging in these activities with unhealthy appearance-focused goals. Secondly, yoga has been shown to promote positive body image, and it's said that this is because yoga focuses on what the body can do and how it feels, rather than how it looks. But what if this trend of idealised yoga gives the message to the average woman that she doesn't belong in a yoga class and undermines any potential benefits to body image and general health? So what am I going to do about it? So next, I'll be using media exposure studies to have a look at how women respond to idealised yoga and how it influences their body image and also their intent and reasons to engage in practice. And I'll contrast that by having a look at how women also respond to more diverse representations featuring models of various ages, sizes, ethnicities, and physical capacity. So broadly, my study contributes to the literature on body image and social media, but exploring these things in a specific yoga context. But most importantly, my aim is for the findings from this research to provide the Australian yoga community with some recommendations on how we can promote and facilitate body diversity in yoga from social media to the mat. Thank you.
Thanks very much, Angela. Our next presenter is Chang Tren, and Chang is going to present to us on the topic of genetic and genomic application for long-term sustainability of prawn farming. Important for any Australian who's into barbecues, I understand. Chang comes to us from our Gynecology Research Centre, based in the School of Science and Engineering. Please join me in welcoming Chang Tren.
Let me ask you the question. Is there any one of you who love prawn? Imagine when we go to the supermarket to buy prawn for a wonderful Christmas party, but there, all the prawn run out or the price is incredibly high due to disease. However, consumers like us not only one who bear consequences from prawn disease. Look at the farmer I have shown over there, how miserable he was when he lost all his property just in the short period of time. White spot disease is a name I suppose so many of you here have heard about that before. The disease is widespread all over the world, and recently it's caused severe losses in prawn production in Queensland in 2016. There are several treatment methods being applied. However, all of them fail to fight against the disease. So, what is sustainable methods to be used to increase the survival rate if the disease occurs? That's also the question I would like to answer. Based on the advances of genetic breeding selection, we attempted to create the new population of prawns that are able to resist the disease, also have the high survival rate in farming conditions. We use the viral copy, the viral methods to select the highest resistant families and then conducted crossbreeding within them. Interestingly, we found that there are more than 12% of higher survival in selected families comparing with the unselected ones. In another effort to find out the new method for breeding selection against the disease, we try to determine the genetic variation in the viral copies in infected stream and also the resistant genes in prawn genome. It succeeds, those approaches can be replaced for costly and time-consuming challenging tests that we usually do before. So our findings contribute to increase the high-quality seeds for prawn farming. It will maximize the productivity for farmers and bring all the happiness for the many lives. And for someone who loves prawn like me, we don't need to concern about the price anymore. So every time we should enjoy our succulent prawn dishes on the table whenever we want. Thank you very much.
Thank you, Chang. Our next presentation comes from a student who's also based in our Gynecology Research Centre in our School of Science and Engineering. Nhu Minh Tran is going to present on the topic of malt biomanagement for a better world. So please join me in welcoming Nhu Minh Tran.
Food safety and environmental pollution are major concerns in the modern society. The global increasing of population requires smarter ways to meet the demand of more food without effect to the environment. My research is applicable into measure of food productions, reduce the effect of insects on agriculture, and enhance crustacean aquaculture. In agriculture, almost 20% of total crop production around the world is lost to insects. So, insecticides were widely applied to protect the crops for a few decades. But the nasty chemicals used have a negative impact on human health and threaten on other life forms. In my study, I focus on the growth mechanism in insect and crustacean. As you can see on the photo to the left, both groups of animals have to replace the old shell in order to grow. And that process is called molting. In my research, I focus on the raw mechanism that can regulate the mold. I have identified a few key molecules that can induce or inhibit the mold. Unlike currently used insecticides, these molecules are natural, biodegradable, and species-specific, which enable us to develop a new eco-friendly approach for regulating the mold in what I call mold biomanagement. I am to use the approach to prevent insects from growing on the crop field. So by inactivating the insects for a certain period, instead of having them to be killed, the food chain in the crop field will maintain its balance and ensure the safety of food supply for humans. I also aim to increase the benefit for aquaculture by regulating the mold to suit the needs of farmers. Instead of having the shrimp molting when they want, we will get them to molt whenever we want. In summary, we aim at mold biomanaging for a better world. Thank you.
Thanks, Newman Tran. We move from some of our smaller animals to a larger mammal in this next presentation. Sarah Pye from our School of Creative Industries is going to present to us on her research around saving sun bears. Please join me in welcoming Sarah.
The research I'd like to introduce you today will be submitted as part of my Doctor of Creative Arts. I'm exploring how a narrative, non-fiction biography can be written to engage a non-specialist audience in conservation. As you can see from this photo, sun bears are insanely cute. They're also at risk of extinction. Sun bears share their habitat with orangutans, but they're known as the world's forgotten bear. Dr. Wong Soo Tee, or Papa Bear, has dedicated his life to saving sun bears, and he's known as the world authority. I first met Wong in 2012, and I was captivated by his commitment and his charisma. I asked Wong, what can I do to help? Do what you do best, he said. Those 5 powerful words have led me on a 7-year journey. I've interviewed over 50 people from around the world. I've conducted an ethnography in Borneo, and I've written a book-length narrative. In this hero's journey, Wong comes face to face with angry grizzly bears in Montana. His car is overturned by disgruntled elephants in Borneo. He tragically loses his field assistant in floodwaters, and he's eventually named a CNN World Hero. I integrate my own story into the narrative, acting as an interpreter of science and culture, and the story ends when I join Wong to release 2 rescued sun bears back into the wild. People are increasingly aware of how we are impacting our environment, which makes this story timely. It uses the lens of one man's extraordinary life to illustrate how humans have impacted our environment, but it also offers hope and acknowledgement that one person can make a difference. My book will actually be published next year, which is very exciting, and Dame Judi Dench has offered to write an endorsement. So this is what she said. Wong is an incredibly committed and charismatic man, and I highly recommend this inspirational story about his life and important work. Thank you.
Thank you, Sarah. And I'm delighted that we've had somebody present from our Doctor of Creative Arts. Many of us across the university continue to refer to our doctorate with the singular PhD, but we do have 2 doctorates here at the University of the Sunshine Coast. And the Doctor of Creative Arts is something we can all be very proud of. What it reminds us is that there are a variety of ways that we can increase our understanding of the world around us. Science is certainly one, but narrative, poetry, art, ways that we inspire and change people through our words, through our pictures, through our creativity, is something that is a worthy pursuit as an HDR student as well. So thank you, Sarah. We change gears for our final presenter and go back in some ways to looking at prawns again. Tuan Nguyen is from our Gynecology Research Centre, and they have a focus in this area in our School of Science and Engineering. And Tuan's presentation is entitled Creating More Prawns for Your Next Barbecue Through Computational Biology. Please join me in welcoming Tuan Nguyen.
Everybody loves prawns on a barbecue. I guess so, right? However, I'm going to tell you a story that you might not know about. In reality, prawns are notoriously for being very tough to deal with. One particular problem is that a lot of female prawns are stuck at a particular point in their life cycle. They're just simply stuck. They cannot proceed themselves to spawn more baby prawns. What happens? Previous study in the past has proven that there are several inhibition factors in the eye stalk of the prawns. The organs similar like the eye of the human. These inhibition factors somehow prevent the prawns from developing their ovary and spawning. Human, very creative, has proven a way to fix this problem by a method called eye stalk ablation, which literally means cutting down the eye stalk of the prawns. Therefore, all of these inhibition factors are now vanished, and therefore the prawns can keep growing. The method is obviously very cost-ineffective, labor-intensive, and at the same time cruel. So my PhD thesis set out to use state-of-the-art computational algorithm as well as high-end biotechnology in order to address these issues. We target a specific group of proteins called neuropeptide, as on screen here, which is basically the protein in the central nervous system of the prawns. These neuropeptides are also known to bind with another group of proteins called G protein coupled receptor, or GPCR. They work together simply like a key and lock combination, where neuropeptide is the key and GPCR is the lock. They bind together and control regulation reproduction. So my PhD thesis has only 3 steps. The first step, which we already did, is to identify all the neuropeptide and GPCR in prawns. The second step, we manually curate them, looking at them one by one, and trying to pinpoint those that perhaps relating to reproduction. And the final, the ultimate step, is to manipulate these, and perhaps in the very near future, we can hopefully completely eradicate eye stalk ablation. And that, ladies and gentlemen, is the story how I use computational algorithm and biotechnology to bring more prawns to your next barbecue party. Thank you.
Thank you, Tuan. I'm sure you'll agree that for these 8 presentations, we have some outstanding talent here at USC. I'd also like to recognise the other HDR students who are attending today who chose to participate in the 3&T competitions and our heats. You are also worthy of our recognition and congratulations. So thank you for taking this on and taking the challenge to present your work in such a brief period. Because it's only 3 minutes, this presentation, the students have not had the opportunity to acknowledge and thank those who support them, and so I'm going to do so on their behalf, if that's okay. What each of them recognise is that the work that they do cannot be achieved without the support of many. Some of those supporters are personal, their family, their friends, their spouses, sometimes children, parents. They also are supported by a team of supervisors. Some are based here at USC, some external. They are also often supported by scholarships, grants, and other activities and actions and materials and lab spaces and opportunities that are created for them to do the work that they do. They're also supported by other HDR students. The community of students we have are extremely important as each of us go through to try and support the journey through to the outcome of wearing that funky gear and going through graduation, which all of us look forward to. So on behalf of the 8 here and the other HDR students in the room, thank you for those who are involved in supporting these projects. Your work, your support is very much appreciated. It's now time to do what I'm glad I don't have to hand over to the judges to spend some time deliberating and making a decision as to who the runner-up and who the winner of this final of the 3&T competition at USC for 2019 will be. Our 2 judges have had a very brief period of time, but they were given some marking sheets with criteria that you will have seen them scribbling notes down on and making calls as the presentations went on. So I'd like to invite our 2 judges to come up to the stage now as I move to announcing both our runner-up and our winner for our 3MT competition for 2019. So can I ask our judges to join me on the stage, please? Thank you to Mike and to Roland for their work in doing this, and I will just hand each of them a certificate to hand out, including the important one, which is the one that's got some numbers on it with a dollar sign in front of it. And I'll just ask you to keep this facing you for now. They have good eyes, these students in the front. So on behalf of the judging panel, it's my pleasure to announce that the runner-up for our 3MT competition for 2019 is Angela Hinz. And on behalf of the judging panel and the University of the Sunshine Coast, our 2019 3-minute thesis competition winner is Chin Dam. I'd now like to invite our Deputy Vice-Chancellor, Research and Innovation, Professor Roland DeMarco, just to say a few words.
So I guess first of all, as the DVCR of the university, it was a great celebration today of the diversity of research we have at the university. You know, everything from the pointy-headed academic science work in the laboratories of genealogy here to the creative industries work that's being done at the university. So you should all be very proud of yourselves. You did a fantastic job. It was very, very difficult for Mike and I to split hairs over who was the winner and who was the runner-up. But I guess just some generic sort of comments from my own perspective. What I was looking for was people connecting with the audience, making their topic come to life in terms of the things that relate to the average person. We're average people out there sitting in the audience, listening to your talk. We're not experts in your domain of research, so it's connecting with us with your topic to make it relevant to us. Very engaging with a good use of sort of quirky ways of presenting your topic, use of humour, that sort of thing. And then obviously in a very clear and concise way which is understandable to the layperson. This is pitching. You sort of explain your topic with sufficient depth but make it understandable, where someone like me as a physical chemist can understand your topic. And so they were the things we were looking for. It was very, very close. Like Mike and I went upstairs there and it's quite a difficult task to pick a winner and runner-up from a group of presenters like today's presenters. Congratulations to each and every one of you. And over to Mike, if you want to add a few thoughts.
No, thanks very much. But look, I think I said a few words earlier in terms of enthusiasm and motivation and passion, and that was absolutely reinforced by by all of you this morning. For me, you all scored very, very highly on the criteria I weighed in on. It was really splitting hairs. And but look, I just wanted to congratulate all of you for your contributions this morning, and wish you all the very best with your endeavours as time moves on. Cheers.
Thanks to you, Mike and Roland, for that difficult job, but especially thanks to you 8 students who've really taken this challenge on, done the work, done the practice, and come up with things that have inspired us all and have helped us grow and learn about your area of focus and expertise as well. As a university, we are a community of scholars, and so the contribution to that scholarly community comes through the work that you do and your ability to share that work with others. So congratulations for taking that on. Thank you for doing so, and all the best for your continued studies towards your degrees, whatever you're aspiring to do. Finally, I'd just like to acknowledge and thank the team who's put this event together today. My team and the Graduate Research Office are a very hard-working group. This is one of the great events that we run. In particular, I'd like to acknowledge Marnie Greer, who's not here today because she's at lectures at the moment, but she's done most of the hard work. Also to Francesca and also to Sally and also to Alarina, who are there, who've done the work to make this event happen today. So please join me in acknowledging that team from the Graduate Research Office.